welcome to Cell and Gene Live. As Miles said, I'm Erin Harris, Chief Editor of Cell and Gene. Thank you for your time today. And joining me for today's presentation are Raymond Luke, Senior Director of MSAT at Verismo Therapeutics, and Todd Luman, Executive Director of Process and Product Development at Allogene. Both of our experts have extensive experience driving innovation and operational excellence in manufacturing science and process development for cutting-edge cell therapies. Now, Allogene specializes in allogeneic cell therapies, while Verismo focuses on autologous therapies. So we have the good fortune of being able to have both perspectives on how early manufacturing decisions are shaping long-term successes. And throughout our presentation, audience participation is central to our discussion. So please do submit your questions at any time using the Q&A tool. We will address your questions as we move through our conversation, so your insights and interests will help shape our discussion today. And I would also like to thank Thermo Fisher Scientific for partnering with us on today's presentation. And we have a lot to get into, so let's jump right in. So with all of our Cell and Gene Lives, I really like to set the stage and level set the conversation before we really dive deep into the details. So Todd, I'd like to start with you. Cell and gene therapy developers need to move quickly to bring first-in-human products to patients, but those early decisions can have long-term consequences. So as it applies to allogeneic therapies, how do you and your team balance the urgency of early stage development with the desire to minimize disruptive process changes in later phases?
Yeah, that's interesting to ponder on. When I think about that question, the executive suite often ponders on how much to invest in a new asset. But what I would ask the audience to consider is thinking about cell and gene therapy perhaps in a single cycle development paradigm. And the reason for that is, it's very difficult in the cell and gene therapy space or cell therapy space to make pivotal process changes due to comparability considerations. So being thoughtful, on where to make the right investment early on can pay extreme dividends later in the development cycle and time and cost.
Yeah, that's a really good point, especially as it applies to both time and cost, which we will, of course, get into throughout the rest of our conversation. Ray, I'd like to hear from your perspective how you design a process that is flexible enough for the uncertainties of early phase trials, but also robust enough to support later stage trials all the way through to commercial production, but in an autologous setting.
Yeah, I mean, unfortunately, there isn't a crystal ball that will tell you exactly what you need in the future for your product or your process. So you have to be as, Nostradamus as possible. You need to be able to try and predict while maintaining some level of flexibility. And how I go about that is you have to build it simple initially, right? You have to be able to have it so that you don't build in a lot of complexity from the start, because building in a lot of complexity from an early phase perspective can be costly, time consuming, and can reduce future automation potential and scalability. Another thing that I always harp on, probably more than a lot of people like to hear, is to collect as much data as possible. In order to make informed decisions about your product and process, you need to make sure that you understand it, and you cannot do that until you can't do that if you do not collect the data. And then, of course, one of the most important things is to stay abreast of innovations in the market. You can't make good decisions if you don't know what's there and you're only working off of old information.
Sure, fair enough. And data analytics, especially, which we will get into also in our talk. But speaking of good decisions and making specific decisions early, Ray, I suppose we can keep you here and ask you this question, because I want to move on to how we're going to align GMP and tech transfer needs. So speaking of early decisions, they need to, or you and your team need to consider GMP expectations and eventual tech transfer, like we said. So
Mhm.
what are those key elements that you're focusing on when you're aligning early development with the long-term demands of commercial manufacturing?
Of course. I think, obviously when you're in early phase, there are phase appropriate differences that you can make. And I think initially one of the most important things to do is to assess the risk of everything that you do. That doesn't necessarily mean to stop, but it means that you understand what the risks so that risks are for future commercializability so that you can then lay out a map so that if and when you are ready to commercialize and this product is efficacious enough and is marketable, you can then push it forward and then you can work on those risks. I think one of the most important things that a lot of companies forget to do when it comes to making sure that a tech transfer will be successful and much easier to go into the clean room is to remember who the end user is. Oftentimes these processes and analytics are developed in PD, process development, analytical development labs, staffed by scientists with PhDs and years of experience. But in the end, this will end up in clean rooms, who are largely staffed by early career individuals who only have a couple of years of experience. So making sure that your process is robust enough is key, and simplicity really is key when you're getting ready to tech transfer, and making sure that you have a lot of what you need hashed out so that there isn't a lot of on-the-ground decisions that need to be made.
Thanks, Ray. And again, to our audience, if you have any specific tech transfer questions aligning with GMP for Ray and Todd, just send them in and submit them, and we will get to them. But in the meantime, Todd, I'd like to hear from you from an allogeneic perspective, of course, those early decisions and what needs to be considered, GMP expectations, and then the eventual tech transfer. What does that actually look like? And what are the key elements you and your team are focusing on?
Yeah, I would echo everything Ray said and expand, and I think it's quite similar in this regard to the autologous, the allogeneic space. What my team does is, as we're developing the process, build out our process description as we go, working with our MSAT folks for tech transfer. And as we get closer to locking this process, start engaging the manufacturing folks and educating around the process along that arc. On the equipment side, as Ray mentioned, automation, closed systems, and maybe we'll talk about this more going along, but ensuring that we have that same equipment on the floor in manufacturing prior to execution, of course, and in PD, and really vetting out that equipment early on to see, especially in the early phases, if it's going to be a roadblock, that could be an opportunity to implement later. And then I would finally talk about this is when you have a really nice process description, work with the manufacturing staff to translate that into master batch records and really pay close attention to the details there. And don't assume anything. As Ray mentioned, we're deep in the process and process development, but you can't make assumptions. So a lot of times the details matter, and how those translate into the batch record and how it works on the floor can be consequential. So really paying close attention there, I think, can add a lot of value.